And we are back to CSEC Bio January 2017. So let's get straight into it. Question 4. Describe how oxygen in the atmosphere becomes available to human blood for delivery to the cells. So let's note that. Becomes available to human blood. And so you're going from atmosphere to blood. That's the direction. Atmosphere, O2 to blood. Okay, good. For delivery to cells. And then you have to explain. So you're describing first, so you're just outlining the process first, and then you're going to explain how it differs from the way plant cells obtain oxygen for respiration. So now we're going to figure out how you're going to do that from oxygen to the atmosphere to plant cells. All right, and you're looking for a comparison. You're making a comparison. So when you see differs, it's a comparison question, right? CXC loves comparison questions, okay? And let's look at what is this question worth? Oh Lord, so much. 9 marks. 9 marks. So I've outlined the answer here in 9 parts. Okay, so the first part you want to talk about describing how oxygen gets from the atmosphere to the blood. So first thing, you get to the blood via, you give a little summary opening, right? So oxygen gets to the blood via the breathing process carried out via the respiratory system. Yeah? So you give the whole summary one time. And now you're going to walk through the process. So first, air is going to move into the lungs via the trachea during inhalation. And then you describe inhalation, right? What's happening during inhalation? What is the respiratory system doing in order to facilitate inhalation, right? So you indicate, okay, the volume is increased, right? The volume of the chest cavity is increased via contraction of the diaphragm, one, and of the intercostal muscles, two, right? What does that do? It lowers air pressure, air moves into the lungs, yeah? Then, and that is your description of inhalation. So notice we're zooming out, we take the big view, and then we're zooming in, right? So first the big view is, well, look, this is how it gets into the blood via breathing and the respiratory system. And the respiratory system. Those guys are working together to think. So let's see. Then we have how does breathing work now? Okay, well, first, how you get it into the body. First, you have inhalation. Air moves into your lungs. All right, and then you describe what inhalation does. How does your respiratory system do that? So that's 3 marks there already. Okay, then you will indicate, okay, when it gets into the lungs, it's going to move to the alveoli. You describe the path, right? And this is one way to describe the path from the trachea right down to the alveoli. Okay, you can outline it. Says, okay, once it gets into the lungs, it can go from the trachea to the bronchi, then the bronchioles, then the alveoli. You could say it that way too, right? This was just my own style. So it is moved to the alveoli via the left and right bronchi, then the bronchioles. Yeah? At the alveoli, oxygen diffuses, because this is important, right? Oxygen is going to diffuse into the nearby blood capillaries, oxygenating the blood passing through them, right? So in other words, the oxygen is going to pass from the alveoli into the blood capillaries that are nearby. The blood that's passing through the capillaries, they get oxygenated. And then what happens to that blood? The blood is going to go to the heart via pulmonary vein and pump to the rest of the body. The next part now, in plants, we want to compare. So we make some comparisons. Firstly, we indicate that, look, there's no respiratory system in the plant. All right. Neither transport system with a pump. Okay. So we clarify that first thing. Well, this is what the plant does not have compared to in the human, right? And then we indicate, well, what does it do instead? Well, how does the oxygen get into the plants? It's going to get into the plants via diffusion through the stomata of the leaves. All right. And you can also indicate that it's gonna be that's gonna be its gaseous exchange. So the gaseous exchange is gonna happen at the stomata. Also, you can mention gaseous exchange at the alveoli when the oxygen is diffusing. You can do that as well. All right. Also, some of the oxygen comes from photosynthesis, where it is a waste product. So it also produces a little bit of its own oxygen, which it can then use in respiration as well, right? And that's basically it for your 9 marks, right? 1, 2, 3, 4, 5, 6, 7, 8, and 9. Yeah. So that's your answer. So just 6 reasons why humans who smoke heavily have less oxygen available to the cells for respiration. There are so many reasons, it's actually relatively easy to just find 6. So I've included the 6 here, and there are further details in my model answers book. If you're interested in getting one of your own, you can check the description below. So let's go through the 6, right? Let's get through the 6 ones. Well, firstly, remember smoking. Cigarette smoke, the components, think of it in terms of the components that are in the smoke that's harmful. You have your carbon monoxide, we have the tar, and we have the nicotine. So once you think of it in terms of these components, then it's a lot easier to outline your 6 reasons. So here I started with the carbon monoxide. The carbon monoxide is going to bind permanently to hemoglobin. So it's going to make it unavailable to take up and transport oxygen. So that is definitely going to make less oxygen available. Yeah. Then I tackle nicotine. Nicotine causes blood cells to become narrowed or constricted, and that's going to limit blood flow. And that also will reduce the amount of oxygen getting to cells. Because if you notice, getting oxygen to cells requires a very intimate interplay between your respiratory system and your cardiovascular system. The both of them work very closely together. And smoking affects both, right? Components of cigarette smoke affects both your lungs and your cardiovascular system. So these are the different ways it can affect your body cells access to oxygen. Yeah. Then we have tar. So we're tackling tar now. So tar irritates the lung linings, leading to inflammation, narrowing of airwaves, and that of course will reduce the air that gets the alveoli, and in turn that will reduce the amount of oxygen getting to the cells. Because less air gets to the alveoli, less oxygen diffuses into the cells, and so on, right? Sorry, less oxygen diffuses into the blood, which of course is what's carrying the oxygen to the cells. My apologies, right? So just on that alone, just by tackling those 3 components, you can come up with 3 reasons just like that. So even if you're having a brain freeze moment, you tell yourself, okay, what are the components in cigarette smoke? Right, well, I know it's carbon monoxide, I know it's tar, I know it's nicotine. You must remember at least one thing for each, right? At least one aspect of those components that are harmful. And you just show the connection, you show how that then leads to less oxygen being available. Yeah? So let's go to the next 3. So we're back to tar again. And another thing that tar is going to do is that it's going to increase the production of mucus. And that's going to fill up the airways, right? So that's going to further narrow the bronchioles. Yeah. And that's also going to reduce the air that's get to the alveoli with the effect that we just mentioned. Less volume in the alveoli, less oxygen diffuses into the blood, less oxygen available to the cells. Yeah? Then the fourth one, still on tar. So notice tar is the main character in terms of damaging your lungs, etc. Out of the six, we have one, two, three, four things, ways in which tar is harming you. I mean, it's harming you in all kind of direction, right? So we have chemicals in the tar eventually lead to loss of cilia. And the cilia is that struck those little, hairs that will help to keep those bronchioles clean, right? And now they're not around. So what's going to happen? You're going to have the mucus accumulating, bacteria getting trapped in the lungs. All right? So I want to in the lungs. That was a senior moment, right? This further narrows the passageway, reduces the volume of air. Again, you're just getting so many things are blocking, are interfering with air getting to the alveoli. It's like the alveoli is right there, but all of these different things are happening to interfere with the air getting to the alveoli. And then the next one, which is, of course, that's a long-term effect, right? Leads to lung cancer that can make breathing difficult and painful. So, of course, if I'm having difficulty breathing and breathing is painful, again, less air getting in, less oxygen diffused, less oxygen for the cells. Yeah? And that's 6. And you can get further details in the book. 5 part A, part 1 now. This one is 4 marks. Okay. So this one, you're being told that blood that has been oxygenated in the lungs, right? So we know that it's starting at the lungs, must first be pumped through the heart before sent to all other organs. Describe the pathway the blood takes as it flows into the heart from the lungs. So it flows into the heart from the lungs until it is pumped out to the rest of the body. Right? So that's important. So not just from the lungs to the heart, but also from the heart to the rest of the body. Right? So if I were to make a little note to myself, I'd be like, from lungs to the heart to the body. Right? So I'd have that and I'm clear in my head. Okay, this is the pathway I'm describing. So I'm not being asked to describe the entire cardiac cycle, right? I'm just being asked to describe what we would call, well, it's not even pulmonary, it's part of the pulmonary and part of the systemic, right? So let's see what steps, and it's 4 marks, so four points. You could break it up into four steps, right? So the blood enters the left atrium from the lungs via the pulmonary vein. Boom. Yeah, right? It's leaving the lungs via the pulmonary vein. It enters the heart via the left atrium. And then next point, left atrium, that's going to pump the blood into the left ventricle past the bicuspid valves. Yeah, because and the valves, you need to mention them because they are a very, very important part of heart function. Yeah. Then that's the next step. The third step, the left ventricle is going to pump the blood out of the heart via the aorta. And the blood is out and ready to go to the rest of the body. And you mentioned this is very important. So the bicuspid valves is going to prevent the backflow of blood into the left atrium. And of course, you would indicate while the left ventricle pumps the blood out. Okay, out. All right, so you have that detail as well. Then we have identified two physiological diseases that affect the circulatory system. There are lots, but the ones that we come across at this level is, of course, the hypertension, coronary. All right. You can also include diabetes. Diabetes does affect the circulatory system. It affects your blood vessels and so on. All right. But the ones you want to focus on in terms of priority answers would be these three. Yeah. And then this interesting question, chemotherapy. Chemotherapy is a treatment that destroys both the malignant and the good cells in the body. Explain why a person undergoing chemotherapy would have decreased natural immunity. All right, so explain, so we have to do some describing, we have to include some describing, some definition, etc. So we state here, first we need to say what natural immunity is, right? So natural immunity is comes from prior exposure. So this has to do with your active immunity, right? Your active immunity. So you get it from prior exposure. The body's lymphocytes develop antibodies against said pathogen. And I know it's they have passive immunity as well, but this is an adult. They're not breastfeeding. Okay. Passive immunity is natural passive immunity is either in the womb or while you're breastfeeding. This person, we can assume, is not of that age, right? So we can just treat it as a natural active immunity. Okay. And then we have state that lymphocytes would have developed antibodies, right? So we don't want to go into long tirade about what immunity is. We just want to give a short summary of what is required. So, prior exposure, you have antibodies developed against the said pathogen. And what does being naturally immune means? It means that your system now has memory cells that can trigger a more rapid, more intense response to the same pathogen on future exposure. So that's what being naturally immune is, right? So let's see what chemotherapy does. So chemotherapy would destroy many of the lymphocytes, especially your memory cells. Because remember, it's destroying both bad and good cells. Huh? And therefore, that is going to destroy the immune system's capacity to deal with pathogens. And even this part, you don't have to write all that. But yeah, you just indicate, look, chemotherapy is going to get rid of a lot of these cells. All right? And it also can destroy other cells that are important for immunity, right? Especially cells that divide regularly, often, right? So your phagocytes could be affected. Your epithelial cells that produce cilia in your lungs, they could also be affected. The acid in your stomach, the mucus, all of these could be affected. And all of these are part of your natural immunity, right? Because your natural immunity also includes those first lines of defense, your skin, the mucus, the different secretions that are produced in any space that is in direct contact with your environment, right? All of these are your first line of defense against any entry of pathogens. Your first immunity is blocking the pathogens from getting in at all. So chemotherapy is going to also be destroying a lot of those cells as well. All right. And that is also going to have a huge impact on your immunity. Then we have Peter, who has not had a tetanus vaccine, gets his foot punctured by a nail. He's given an anti-serum instead. All right. So we know anti-serum would be your artificial passive immunity. Okay. And they ask, well, why not just give him the vaccine instead of just giving him this anti-serum, right? Because the vaccine will make him immune, right? Well, the problem is timing, right? So the first thing indicate, okay, this is how a vaccine works. All right, so it's going to work by triggering your primary immune response, and that takes a while to be established. Okay, that's why you take a vaccine, to get past the blow past the primary immune response. That usually takes a lot longer versus the secondary immune response, which would take a lot faster. So it takes a shorter time, I should say. So you want to make sure that if you do encounter the pathogen in real life, like really encounter the pathogen, you're not having to first go through that initial primary response. That's going to take a while and going to allow the pathogen to establish itself. All right. So that's why you take a vaccine, to get past that primary immune response. But it means that it also means that, yeah, it's going to take a while to work. Yeah. And therefore, that's why it's better you use it before you get infected. All right. And we indicate that tetanus works really fast, right? And it's going to work much faster than the primary immune response. And it works fast to the point of lethality. It works fast to the point of causing major damage before your primary immune response can step in. So therefore, it makes more sense to just give you the antiserum. All right? So tetanus acts faster than the primary immune response can work to produce antitoxins. By using the ready-made antiserum, it's more effective because once infection has already occurred, the antiserum can quickly neutralize any toxins already produced. Yeah? So that's basically how you would put your answer together. And as again, notice they give you enough space so you can take your time and explain your answer properly. I usually suggest, part of the reason why you want to conserve your time in questions 1 to 3 is so that you can really take your time in questions 4 to 6, even to the point of writing it in pencil first, doing a draft on the side first, draft out your points on the side, and then write it out like a mini essay. And now for question 6. Describe how genetic engineering is used to produce human insulin using the bacterium E. coli. And this one is worth 6 marks, so 6 points, right? Or 6 steps. So first step, to one section of DNA that codes for human insulin. So basically the gene for human insulin is extracted from human cells, or you can synthesize it using this particular enzyme. Yeah. Then we have DNA strand is added to plasmids from E. coli bacteria. Right. Remember when we talked about the bacteria in part 1? So you have a bacteria, you have a nucleoid, and then you have these tight loops of DNA. Those are your plasmids. Very useful, guys. Right? So these modified plasmids, they're inserted into E. coli bacteria. Right? You're going to take those plasmids, open them up, add the DNA strand to it, close it up. Now it's a modified plasmid, and you're going to insert that into other E. coli bacteria. Then the insulin is going to become, well, of course, now the insulin is part of that bacteria's genome, right? And now that bacteria is officially known as a genetically modified organism. GMO. Organism. Right? GMO. Right? And then we have the modified E. coli. They're allowed to multiply in a fermenter. So basically allowed to multiply and grow. And while they're doing all of that, they're going to be producing insulin and producing a lot of it because they don't need it. And there's no real regulatory anything added to it. So it's just going to be pumping out insulin because based on the DNA provided. And then afterwards, you shut off the fermenter, and extract the insulin from the contents of the fermenter, and you put it in a little bottle and sell it for 400 and something dollars, which is so sad. Then we have part B, three differences. Now, is, there's so many. In fact, I'll show you how many I was able to come up with. But I included three here, of course. Discuss three differences. It's still a comparison question. Treat discuss as very similar to explain. So you want to look at differences between genetic engineering and artificial selection, right? So artificial selection would be what you would know as breeding, all right? Like breeding a horse, breeding plants, etc. So I gave three. So one is that artificial selection, which is the breeding, you can carry it out in a field or a farm. All right. You don't need a whole lot of expensive, crazy equipment to make it happen. You just need your animals or your plants, etc. You keep them, keep them, and you just need to keep track, keep a record, and all of that. Some in these days they would also include some artificial insemination, especially for like cows and so on. But still, it is a relatively emphasis on relatively low tech strategy as compared to like genetic engineering, which you need a specialist controlled environment, right? Specialist controlled environment. You need a lab. Just imagine people walking around with their lab coats and their face covered with masks and all the craziness, right? So that's one difference. Another difference is that to get the variety that you want, get the features that you want using breeding, which is your artificial selection, it's going to take longer. It's going to take several generations. Yeah. Genetic engineering, it goes faster. Yeah. And it's a lot more precise. Yeah, so you're gonna get the desired results within the generation. Within the very generation that you're treating, you're going to get those traits, right? By inserting the appropriate gene, and there you go. You have your results. And a third one is that artificial selection, you're not going to get the exact, it's not as precise as genetic engineering, right? Because again, you're just letting things roll naturally. You are just determining who mates with who and hoping that the genes will mix and match the way you want them to, but you can't really control which genes will mix and which will match, right? So you're going to have desired traits, you're going to have undesired traits mixing up together and being passed on in the offspring. Yeah. And with genetic engineering, you're more precise. You can get more precise targeting. You can focus on exact trait that you want, right? So it's that's a big advantage that genetic engineering has over artificial selection, as well as the speed in terms of generations that you can get it done. All right. There are several other details in the book. I'll just give you a little snapshot of what it looks like. Right. Yeah. So as you can see, it's a lot. All right. It's a lot. Now, of course, if you're just getting ready for the exam and so on, you don't have to put yourself through this torture. But if you're studying the subject, you want to understand it fully, and okay, I'm going to do this at CAPE level, etc. I want to have a strong understanding, then it would be a good idea to get this kind of information available to you. And I can guarantee you, this is not in any one textbook. I had to put this together through research, going through more than one textbook. Yes, I had a little talk with ChatGPT, right? And Bing and double check and all of that to be able to compile this, right? So again, just a little pitch of the value of the book that I've put together. Yeah. And the quality that you're going to get. Yeah. So enough pitching. Let's go back to our video. Let's go back to our past paper. Right. So this is B part 1. So let's go on to the next part because we're almost there. Last part now. So 3 advantages of genetic engineering to produce insulin. So what are the advantages of using insulin that came from genetic engineering? Well, there are many, right? I listed 1, 2, 3, 4, 5, 6. You could choose any 3. All right. You have a shorter space of time. It doesn't take as long. Doesn't take as many resources either, right? Because remember, the alternate is to extract a whole lot of pancreases from pigs or cows and then go through the tedious work of extracting cow and pig insulin from the pancreases before it degrades and then purifying it and then selling it, right? So it's a lot more work, it's a lot more tedious. And even after you do all of that, it's still pig and cow insulin, which means there isn't going to be that perfect fit, right? So as you can see here, you have a large amount of insulin produced more frequently and easily, right? Easier, I should say. Okay, and as compared to getting it from pigs and cows, then you have mass production is cheaper for the consumer. Well, it's supposed to be. That's another story for another day, right? Humans, they don't develop resistance because the genetically engineered insulin is human insulin. It's just that we use bacteria to make it for us, right? So our bodies may treat pig and cow insulin as an outsider, right? Which it kind of is, right? So you could have some bad reactions to the pig and cow insulin. Then you have again allergic responses, right? Less chance of an allergic response. And for those of us, for religious reasons, right, we don't interact with products of pigs and cows. This helps to get around that problem because now I don't have to worry about do I live or do I, go against my religion by inserting, these pig and cow products into my body. Yeah. Now I just use the genetically engineered insulin, right? It's human insulin. Yeah. So that's basically it for that, right? So you have 6 options and you just have to choose any 3. Okay. And that's it for January 2017. Yay!